In this lecture, we are going to dive into the details of reabsorption in the proximal tubule. Take a quick look at this image, which just revisits the 4 processes that occur in the nephron, and number 2 is reabsorption. When we take a look at where reabsorption occurs, we see that it occurs basically everywhere except Bowman's capsule. But what is particularly striking about this image is just how much reabsorption occurs in the proximal tubule. Indeed, the proximal tubule certainly pulls more than its fair share of the reabsorption duties, and even a little bit of secretion happens in the proximal tubule as well. Now you'll see here that the list says nutrients. Remember, nutrients are going to be glucose, amino acids, and nucleic acid, sodium and chloride. We have water, we have bicarbonate, and what's not shown on here are some waste products such as urea. We will not be covering all of those substances, but we will certainly be following some of them. So I am going to draw out the process of reabsorption in the proximal tubule with a special emphasis on how sodium drives everything. We pay for the active transport of sodium, and then we get everything else, or almost everything else, coming over passively. So I mentioned in the previous lecture that there's 2 sayings in renal physiology, and they're used somewhat interchangeably, one of which is sodium drives everything, and the other of which is the sodium-potassium pump drives everything. They are related concepts, and they are both true. So these are going to be the substances that we'll follow. We'll look at water, sodium, chloride, glucose, and urea. So let's take a look, get out those drawing pencils or pens. So to start, we are going to blow up or enlarge a small section of proximal tubule. So if I take that small boxed area and expand it, I'll realize that I have a layer of cells. So these are cells that line the kidney tubule. They are simple cuboidal, and that's going to separate out our drawing into 3 sections. So we have our filtrate on the left, then we have our intracellular fluid inside of our cells, and then we have our extracellular fluid, which is also interstitial fluid. And tight junctions are going to join those cells in order to ensure that substances cross both the apical and basolateral membrane in order to be reabsorbed. Now, reabsorption, remember, is going to be at least a 2-part process. If I have solute X, then it needs to be able to move across both the apical and the basolateral membrane. So both of those substances need to happen in order for reabsorption to be achieved. Now we're going to map out some of our solutes. If you're drawing this with me, go ahead and get out some color like red and start drawing in sodium as shown here. So sodium is going to be concentrated in the extracellular fluid and also the filtrate, because remember, the filtrate is simply plasma that's been squeezed out. So make sure you concentrate your sodium in the filtrate and the extracellular fluid and have very little, if any, in the intracellular fluid. Now we're going to add chloride to our drawing. Remember that sodium and chloride are 2 peas in a pod, and wherever you find sodium, you also find chloride. The concentration of chloride tends to be a little bit less than sodium, but it's still a dominant ion in the extracellular fluid and therefore also in the filtrate. Now we're going to map out our glucose and our urea. The important thing to note is that urea has an equal concentration everywhere. Indeed, the concentration of urea in the filtrate is the same as the one in the cell, which is the same as the extracellular fluid. Glucose is slightly more concentrated inside the cell than outside of the cell. Now we are going to start demonstrating reabsorption. But before we do that, we need to first start with some transporters. And why not start with the one that drives it all, the sodium-potassium pump? So I'm using a black oval to denote the sodium-potassium pump. The sodium-potassium pump is going to be inserted on the basolateral membrane here. So go ahead and add them to your drawing if you are drawing along. Now let's take a look at the sodium and the glucose in the filtrate. Is there a gradient for sodium to come into the cell across the apical membrane? And the answer is yes, there is. Sodium is more concentrated in the filtrate than the ICF, and so sodium would want to come in. But glucose would be going against its concentration gradient, and so would not want to enter the cell. However, we have a solution where we can partner them both up in order to bring them both across at the same time. And that is the sodium glucose cotransporter. So if we use the sodium glucose cotransporter, we allow the energy that is released from sodium coming into the cell as it wants to bring its unwilling friend glucose against the concentration gradient. I'm only going to show that transport in the middle cell here, just for simplicity. So we'll go ahead and add in our sodium glucose cotransporter, but then I'm going to go ahead and show how sodium and glucose are moved into the middle cell only. Notice that we've accumulated 3 sodium ions in our cell. We can pump those sodium ions out via the sodium-potassium pump. So remember that these processes are happening concurrently. So let's go ahead and take a moment and just move the sodium out of the cell. So now I resume transporting my glucose and my sodium together across the apical membrane with my sodium glucose cotransporter. At this point, I have 2 glucose molecules left in the filtrate, so I can do this transport 2 more times before I run out of glucose, and therefore I can no longer use that transporter. Look at all the glucose that's accumulated in the cell. There is now a strong gradient between the intracellular fluid and the extracellular fluid for glucose. I can let that glucose leave the cell passively with a carrier protein, like a GLUT transporter. So let's go ahead and add that in, and then let some of that glucose exit the cell passively. Notice that things are starting to look a little crowded in the extracellular fluid. In fact, let's take a moment and actually count how many sodium ions are going to be in each compartment at this point. So I'll quickly go ahead and count the number in the filtrate, and then I will count the number in the extracellular fluid, and we'll compare them. Notice that there's more sodium ions in the extracellular fluid because we've reabsorbed them. Chloride ions, however, have yet to move. So when we take a look at the chloride ions, there's still a little bit more in the filtrate than the extracellular fluid. But what's particularly interesting is the difference in charge. This extracellular fluid now has a more positive charge than the filtrate does. And what's attracted to positive charge? Negative charge. So what that means is that there is an electrical gradient for chloride at this point. So chloride is not necessarily compelled to move from the filtrate to the extracellular fluid by chemical gradients, but it does have some pull based on its electrical gradient. In other words, it's simply more positive over in the extracellular fluid, and so it wants to go that direction. So we're not going to get into the mechanics of how chloride is reabsorbed. A lot of it is through channel proteins. And truth be told, there is actually a little bit of active transport involved in chloride. However, for our purposes, we're going to assume that chloride movement is passive, that chloride follows its electrical gradient. When sodium moves, chloride goes with it because chloride wants to be where the positive charge is. And certainly that electrical gradient does account for some sodium movement that is passive or free. So we've looked at sodium, glucose, and chloride movement. And one of the things that you should notice right now is that the solutes are really concentrated in the extracellular fluid. So if we look at the osmolarity at this point in time, the osmolarity of the filtrate is going to be much lower than the osmolarity of the extracellular fluid. Guess what follows an osmotic gradient? Water. Water follows. So water is going to follow the solutes. So this is how water is reabsorbed. We transport solutes and water follows. I want you to take a good look at this picture with special attention to the volume of the filtrate, which is shown here, versus the volume of the extracellular fluid. The volume of the filtrate is greater than the volume of the extracellular fluid, but as water leaves the filtrate will get smaller and the extracellular fluid will get bigger. So I can redraw that by changing the size of my filtrate and ECF as shown here. And by changing the sizes of my filtrate and my ECF, I condense the solutes in the filtrate and give the solutes in my ECF more room to spread out, such that the distribution of solutes looks almost the same and my osmolarity is equal. Indeed, water will continue to follow solutes as long as there's an osmotic gradient, until the osmolarity between the filtrate, the ICF, and the ECF are equal. To reiterate, water reabsorption is passive, and water always follows the solutes. So as solutes move, water follows. Now that water has moved largely from the filtrate to the ECF, something interesting has happened with urea. But unfortunately, this drawing is a little crowded to really show it well. So what I'm going to do is I'm going to show you the same drawing with all of the solutes except urea removed. Now you might be looking at this and going, I don't see what the big deal is. But it will help if we outline the various compartments. So let's look at the volume of the filtrate, the ICF, and the ECF respectively. And when you take a look at that, you'll realize that urea is slightly more concentrated now in the filtrate, especially relative to the extracellular fluid. That is because 4 urea molecules are crammed into a smaller space in the filtrate. So urea now has a gradient, and so urea is going to want to move down its own concentration gradient, and so we're going to show that here, where we're going to spread urea out a little bit more so that it shows an even distribution in our green box. So urea also gets reabsorbed passively because urea follows the water. So it's a really interesting setup where sodium moves first with glucose or an amino acid or a nucleic acid, and then chloride follows electrically, water follows all of it, and then urea suddenly is more concentrated in the filtrate because water has left it, and urea follows passively. So that is what we mean when we say sodium drives everything. Okay, now that we've seen the ingenious way in which the sodium-potassium pump sets up a gradient that ends up drawing sodium across, and then everything else kind of follows through various types of chemical or electrical gradients. This image here is from your textbook, and it shows some parts of what I described. It's a little bit messier, but I do want to go through it. You can see here that this is the filtrate. This is the intracellular fluid. Again, tight junctions are going to be used to make sure that substances don't make it through. This is going to be the interstitial fluid, which is just extracellular fluid. In the basolateral membrane, we have the sodium-potassium pump here. This is where we pay for that sodium pumping to pump sodium out of the cell. That creates a gradient where there's very little sodium in the cell, which of course is going to draw sodium into the cell. But sodium is going to bring a friend. It's going to bring glucose with it using the sodium glucose cotransporter. Now, something that I didn't mention earlier is that we use glucose as a stand-in for nutrients, but amino acid reabsorption and nucleic acid reabsorption is done the same way. In other words, we use a sodium amino acid cotransporter or sodium nucleic acid cotransporter to bring those nutrients across as well. Ideally, 100% of all of your nutrients will be reabsorbed in the proximal tubule. So this is total reabsorption, or it should be total reabsorption. The other thing that you might notice here is that we have a glucose carrier. So it says facilitated diffusion carrier here. That refers to like a GLUT carrier transporter that allows glucose to exit the cell passively because it's following its own concentration gradient. The urea scenario is rather interesting, and this image takes a closer look at how urea follows water. In this image, every purple dot is a urea molecule, and what you want to pay a special attention to is how urea is spread out in a particular compartment. So here it's looking at a certain amount of filtrate and it's got a certain number of urea molecules. And if you look at the spread outness of urea, it looks pretty similar basically everywhere, right? So urea does not have a concentration gradient right here. But as you move sodium over and then chloride follows and glucose goes and amino acids go, water's like, where are the solutes going? It's going that way, so I'm going to go that way. And then that dehydrates the filtrate, leaving the urea behind. And so now you can see that urea is much more concentrated here. So now urea is going to follow its own concentration gradient and follow water. So the rules are sodium drives everything. Sodium brings along the friend glucose or an amino acid or a nucleic acid. And as sodium moves, chloride goes, but wait, it's more positive over there. Chloride follows largely from an electrical gradient. And then water goes, why is everyone going that way? I want to go too. And water follows along. And then urea goes, but wait, now I'm all concentrated here and everybody's moved over there, so urea follows. So sodium transport really does drive much of the reabsorption that happens in the proximal tubule. In our next lecture, we are going to learn about the really fascinating setup in the loop of Henle. I'll see you then.